Electrophoresis is the separation of charged molecules in an applied electrophile. It is the standard method to separate, identify, and purify DNA and RNA fragments. Some applications of electrophoresis are DNA analysis. It can be used in forensics and paternity tests. Protein analysis, used to measure the quantity of a protein in our body. If the levels are not normal, may indicate a disease. Antibiotics analysis, used for the ability to synthesize new antibiotics and to analyze what kind of bacteria are resistant. Vaccine analysis, because some vaccines have been purified, processed, and analyzed with electrophoresis. To make an electrophoresis, first you have to make the gel. Then you have to add a dye and put it into a container, wet the gel. Then you have to put the gel into an electrophoresis chamber and cover it with buffer. Finally, the samples of DNA and RNA were placed in wells and the voltage is applied. You have to use fluorescent staining.
In this table, we can see the factors that we have to know if you want to make an electrophoresis. For DNA and RNA, is commonly used a polyacrylamide or agarose matrix, with buffer TBE or TAE, using dyes like HealthRed or CyberSafe. When you are working with proteins, the sample visualization can be with a silver staining, Kumasi staining, or fluorescent staining. Finally, you have to soak the gel in glycerol methanol, making a smooth rotation movement. So, in this way, we can see easily the strips in gel.
When you are going to use RNA samples, is required an extra step to denature it before and during the electrophoresis. This is to prevent the formation of secondary structures that affects the migration of RNA. For this process are used glyoxal DMSO, formaldehyde, or methylmercury hydroxide. If you want to see proteins in a polyacrylamide gel, you can use silver staining, Kumasi staining, or fluorescent staining.
This gel have in the first well the molecular weight, in the second a PCR product, in the third a genomic DNA of human, and in the last one a plasmid PROEX. The first gel was at 0.6% of agarose and it was run at 120 volts, 45 minutes with TVA buffer at 0.5x. And in this image we can't see anything in the first well, and in the second the sample is swept. In the next well, part of the sample get out of the gel, and in the last one we can see some isoforms and also part of the sample get out. In this gel, the voltage is too much for the lower concentration of agarose. The second gel was at 1.2% of agarose and also run at 120 volts and 45 minutes with TVA buffer at 0.5x. In this case, we can see the molecular weight, but in the second well, the sample could be contaminated with DNA fragments and also there are too much sample. In the next well, the sample is degraded and in the last well, the sample does not reach out the well. In this case, we could see that more time was needed to have a better image of the gel. The third gel was with 2.4% of agarose with the same conditions. In this gel, we see the molecular weight. In the second well, there are too much sample and could be contaminated with DNA fragment. In the next and in the last well, the samples does not reach to get out. In this case, the time was not enough and we need more voltage. The last gel was made with 1.2% of agarose with TAE buffer, and in this case, the second well, the sample could be contaminated with DNA fragments, and there is too much sample. In the next well, the sample does not reach to get out, and in the last well, we could see the isoforms of the sample. Also, in the B, C, and D gel, we can see two white stains, and this is because of the excess of dye. The first one is bromophenol blue, and the other is helianol.
When running an electrophoresis, it's important for us to know the percentage of agarose or polyacrylamides we're going to need. This is because depending on the size of the DNA is the percentage of the matrix we're going to need and use. When we are running an electrophoresis, it's important for us to know the quality of the DNA extracted and that we are observing in the gel. Through electrophoresis, we can know the concentration and integrity of the DNA, and we can also know the size of the fragment of DNA. Through electrophoresis, we can separate the biomolecule through a matrix, which function is to be a filter for the molecule to be separated.
CyberSafe DNA gel stain is a highly sensitive stain for visualization of DNA in agarose or acrylamide gels. It is specifically formulated to be a less hazardous alternative to ethidium bromide that can be used with either blue light or UV excitation. For the report, an agarose gel with low percentage of concentration and very high voltage was run. As a consequence of this, the samples couldn't be correctly visualized. If it is necessary to increase the voltage, we should consider that the speed of migration of the fragments in the gel will increase proportionally. For the concentration of agarose used in the report, we should have applied a voltage of less than 60 volts.
As we have seen, the percentage of agarose, the size of the sample, and the voltage is important to consider when running an electrophoresis. For example, in the first gel, we saw that we have low percentage of agarose and high voltage, and the results couldn't be seen correctly. In the other hand, in the other gel, we saw that we have more percentage of agarose, the same voltage, and the results could be seen better.
In conclusion, we can say that the 1.2% agarose gel using TAE buffer is the best for the samples that we use in the practice, because with voltage of 100 watts and 45 minutes, the samples of molecular weight marker, PCR product, genomic DNA, and plasmid PROEX were separated without swept, so we can see the strips perfectly. It's necessary to mention that the genomic DNA sample is totally degraded. In the first gel, we put a lot of voltage for the little concentration of agarose, and in the third, maybe we need more time of running, so in this way we can see the strips better defined.